Hi, everyone. Let's go ahead and get started. Thank you, everyone, for taking the time to join the Arthritis National Research Foundation's second in a 5-part webinar series. I'm Emily Boyd-Suurmond. I'm the CEO of the ANRF, and along with our board of directors, staff, and those participating today in the panel, I'd like to welcome you to the Spotlight on Lupus. We're so grateful for your participation and support and appreciate your interest and time today. Moderating the discussions is an esteemed member of our Scientific Advisory Board, Dr. Betty Diamond. Dr. Diamond is a professor and head of the Center for Autoimmune and Musculoskeletal Diseases at the Feinstein Institute for Medical Research. Before we start the webinar, 2 quick logistical pieces of information to know. Number 1, this webinar is being recorded and we'll have it available on our website at a later date. If you have any questions, please type them into the Q&A and we'll try and get to all of them. If we aren't able to get to yours, please email info@curearthritis.org after the webinar, and we'll make sure to answer your question as well. Now let's move on to the presentations featuring the impressive work of our ANRF scholars. Without further ado, I'm happy to turn over the webinar to Dr. Diamond.
So it's a real pleasure to host this session on systemic lupus. As you know, the ANRF has an extraordinary record with respect to identifying outstanding talent for fellowships, and you'll hear from both alumni and current fellows of the ANRF who are studying various aspects of lupus and who are the future of lupus research. So we're going to begin with a current fellow, Maria Gutierrez-Arcelus is originally from Mexico. She received her Ph.D. from the University of Geneva in Switzerland and performed a postdoctoral fellowship at Harvard. Since 2019, she's been an instructor at Harvard Medical School, and she's studying the processing of messenger RNA transcripts in lupus. So Dr. Gutierrez.
Thank you, Dr. Diamond. Thank you very much for the introduction. I'm going to share my screen. Can you see my slide? All right. Great. Well, yes, Dr. Diamond said, I'm Maria Gutierrez-Arcelus. I just started my lab at Boston Children's Hospital earlier this year in systems immunology and regulatory genomics to understand immune-mediated diseases. And I'm very interested in understanding the pathogenesis of lupus. So today I'm going to talk to you about alternative splicing. We have a gene here on the top that has exons and introns, and normally this gene gets transcribed. So you have a pre-messenger RNA with the exons and the introns there, and then through a process called splicing, you normally this gene gets the introns spliced out. And what is very interesting is that through alternative splicing, we can get different versions of the same gene. So in this example, the 3 exons were kept in this version of the gene or this isoform. And in this other version, the blue exon in the middle got spliced out. So these 2 different versions of the gene can give rise to different proteins that can have different functions. This type of phenomenon occurs in over 90% of human genes. And here's a simplified version of 2 versions that come out from the same gene, but this can have a single gene can have 7 or 10 versions of its gene and give rise to different proteins. And this is a part of gene regulation that has been understudied compared to other sides of gene regulation. So I think it's important that we pay more attention to it and we can take advantage of public data sets to analyze this phenotype, alternative splicing. So alternative splicing plays important roles. One of them, it can influence cell lineages. You can imagine if this gene is producing more of this version of the protein, you can give the cell different a particular set of functions that help become this cell type, whereas in a different cell, you might be producing different kinds of proteins maybe from the same gene, and this can give rise to different functions and give rise to a different cell type. Alternative splicing can also influence cell states. So maybe you have a resting cell here, and then it gets activated, and different versions of the same genes get produced, and this can give rise to different functions for this same cell type. So here we're very interested in studying alternative splicing that influences B cell states in lupus. And one approach is comparing, for example, here, healthy controls on patients, where patients we might observe a different splicing pattern in their cells, and this can give us clues as to what is happening in the disease. And why B cells? Well, B cells are known to be very important for lupus, and in particular, genetic studies have given us a lot of this information of how B cells are very important for its pathogenesis. So I'm going to talk very briefly about the genetics of lupus. Genetic studies have found hundreds of risk variants that give us susceptibility to lupus, and these variants tend to be of small effect. So if you have this risk variant, you increase your likelihood by just a little bit of getting lupus. And these variants tend to be non-coding or outside of genes. And it turns, so this makes it more difficult for us to understand how these variants are leading to disease, because if they were inside a gene, we would be able to study more directly, okay, what happens to that gene. But in this case, they're outside of genes, and studies have shown that these risk variants are enriched in regulatory elements that are specific for activated B cells. So we think that these variants are affecting gene regulation in B cells during their activation, and this can lead to the accumulation of these risk variants affecting gene regulation in B cells can affect B cell states that then lead to intermediate immunophenotypes that then lead to the disease. So the better we get at understanding these mechanisms, the better we'll get at designing new therapies. And the beauty about genetics is that you can assign a causal direction of effects. So if you see an association between a genetic variant and disease, or a genetic variant and a molecular phenotype of a gene, you know that there's some change in the genome that is causing that phenotype that you're observing. So thanks to that causality that we can infer, also if we have a new drug target that arises from a genetic association study, those drug targets that have genetic evidence tend to be more successful in clinical trials than those drug targets that don't have genetic evidence. So we're interested in studying also these genetic effects, and the big challenge here is that for most of the risk variants for lupus, we haven't found the regulatory effects. So there's the big problem of the missing regulatory effects. And one possibility is because we haven't yet ascertained all the B cell states systematically. But another possibility is also that we haven't been analyzing all of the different phenotypes that you can analyze in gene regulation. And one of them is alternative splicing. So we want to identify genetic risk variants for lupus that are affecting alternative splicing in B cells. So that's summarizing these two independent approaches of how we're studying alternative splicing in lupus. One is comparing patients versus healthy controls, and the other one is looking at the genetic effects of risk variants. And the way we are approaching this is by reanalyzing public data sets to make new findings and then validate these new findings experimentally. And to just give you a quick taste of the type of things we're finding, this is just an example where we reanalyzed this data set from Sharer et al., where they isolated B cells from 9 SLE patients and 12 health controls. And we reanalyze this data set with leafcutter, which allows us to quantify these links between exons. So you can see in this gene, EIF4A2, in healthy controls, 71% of the links between exons occur between exon 1 and exon 3. Whereas in lupus patients, this occurs only 21% of the time. And in lupus patients, you have more links between exon 1 and exon 2 and exon 2 and exon 3. So you have a larger inclusion of this middle exon. So that's it for today. I would like to acknowledge Jing Chen, who is the one that did this analysis that I showed you. Victor Nieto in my lab is also working on the genetic aspects of splicing and lupus. And these are other members of my lab that I'm sure will contribute soon. They're just starting. They will contribute soon to this project. I would also like to thank the Nigrovich lab. Peter Nigrovich and Qiang Wang have provided preliminary data, and they're fine mapping lupus genetic variants, and it helps us also think about how this could be causing splicing disruption. Thank you to the patients and healthy volunteers that give samples and allow for their data to become public. This is very valuable. And also thank you very much to ANRF for funding this project. We're very grateful for that. And I can get any questions. Thank you for your attention.
So I actually have a question. We're used to thinking about these regulatory sequences in genes as regulating the amount of transcript that's made, as opposed to the nature of the transcript itself. And so I was wondering if you had any idea about how often the regulation is going to be of the amount versus how often it's going to be of the actual transcript.
That's a great question. we haven't really quantified that. I know there's some groups that are working on that quantification to understand, how much is it that these variants are affecting how much a gene is expressed, or which version of the gene you're expressing. We don't know yet, but definitely it's important to quantify that. And I think it's more than we expect, because most people, only when they see a variant inside the gene in an exon-intron junction, do they suspect that maybe it's affecting splicing. But in my PhD, we were studying how these genetic variants are affecting alternative splicing, and we saw that these genetic variants were also enriched in enhancers, which are distal regulatory elements. So we think that these known coding variants, a lot of them could be affecting splicing, and we haven't systematically ascertained that yet.
Can I ask one more question? So we know that children get lupus and that adults get lupus, and that pediatric lupus is about 20% of lupus, and it's a more severe disease. Do you think that this phenomenon occurs more in pediatric lupus or more in adult lupus? And I'm asking really whether you think this phenomenon is developmentally regulated at the level of the whole organism as opposed to the individual cell. So do you think this will be more common in children or less common in children, or will be unaffected by the age of the patient?
Yeah, that's a great question too. I don't know the answer, but we do know that lupus is more severe in children, and studies indicate that children have a larger number of the lupus risk variants than adults. But yeah, it will be very interesting to see if not only the number of risk variants, but maybe the type of risk variants. Maybe, yeah, maybe children have more of these splicing variants that could cause more extreme phenotypes. In like, if you modify the level of expression of a gene, you could imagine it might be like different, less extreme than if you're modifying which protein you're actually producing, right? So yeah, it's something that we can definitely would be great to ask in the next few years as we analyze more pediatric lupus patients, which I'm already involved in a big project on that, so I'm really looking forward to that.
Okay, so are there any other questions?
No other questions at this time, but we'll continue to monitor the Q&A and we'll let you know.
Okay, so maybe we should go on to our next speaker then. And our next speaker is Tim Newald, who is the most senior of today's speakers. He received his MD from the University of Illinois and his rheumatology training at Cornell, where he was involved in pioneering studies of the role of interferon in systemic lupus. And he's currently a professor of medicine and pathology at NYU Langone Medical School. I should just say that one of the most recent drugs that's been approved for treatment in lupus blocks the interferon pathway. So it's work that is initiated by ANRF fellows that really can, in an exceptionally short period of time, in truth, translate into improvements in clinical practice. So Tim?
Great, thank you. Thanks for that introduction. Let me share screen here. Sorry, it's just taking a second here. Okay, can you see my slides okay?
Yes.
Yes, okay, great. Thanks. I had a little technical difficulty. I'm on the iPad, but I think this should work. So thanks for that great introduction. And yeah, the ANRF was an early supporter of my career, the first actual external funding agency that supported me. And so I have a really a place in my heart for the ANRF. You know, it's a time when many of us are hoping to launch and it doesn't seem very certain that you're going to make it or not. And so the ANRF was there for me. And it really was a great sort of boost in my early career. So I'm very happy to speak to everyone today. And I'll just kind of give a high level of some of the things that we're interested in now. This slide on lupus pathogenesis, I think we you know, some of the other speakers already alluded to many of these pieces. You know, there's, and I just wanted to sort of plug in some of the areas in my current program where we're trying to address some of these features, just following a nice presentation on genetics. And we think that between the genetics and the environment, you really lead to the cellular immunity and autoantibodies and preclinical autoimmunity seen in the disease. So, you know, what are we doing? I'm just going to give some thumbnails of a few projects in the lab now that are addressing different parts of this. My initial work has been in genetics and genetics of the interferon pathway, and that's something that ANRF helped support early on for me. And we've continued with that. We think that that's an important way that you can, as a previous speaker said, you use a directionality to it, and you know that the genetic variants are causing disease. Many times there's a big black box. You don't know how the genetic variants are causing disease, and that's unsatisfying for some people, but I've been okay with that personally, and we've been sort of trying to push into that black box of how do you go from a gene variant that comes out of a genetic mapping study all the way to disease pathogenesis, because we know unequivocally that some of these things are linked, and so it's sort of the next work to fill in the blanks in between about how the gene actually causes disease. So the first project here with the arrow, this PNP project, I'm indicating is one that we've pushed ahead with an interferon-related gene that has been interesting, and I think is going to help unravel some of the complexity in this pathway. As Betty had just mentioned, that we're really excited that there's an interferon targeting drug. It looks like many of our lupus drugs, it's going to work for some people and not for others. And I think there's still a lot of diversity in the lupus population we need to understand. So we feel that the work on these interferon-related genes is just as important now, even with a therapeutic that's out, because I think different people's pathways are tuned in different ways. As another strategy, trying to target some of the interferon-producing cells, the cell that looks kind of like an octopus with a DC label on it, we intend that to be a plasmacytodendritic cell, which is one of the main cellular producers of type 1 interferons, at least as far as we know. I think there are other producers, and it's been an interesting active area of research to try to understand where the interferon comes from in lupus. I definitely don't have time to get into that. I think the short answer is we're not sure, but we think the PDCs are doing at least a good portion of it. And so we have a project looking to try to shut down plasmacytodendritic cells potentially. And then after you start generating an immune response and make autoantibodies, we know that many people generate these autoantibodies before they actually get sick. And so we've been interested in the autoantibodies as an early pathogenic feature. And we've done some genetic mapping that's led us to be interested in this IRF5 gene and how it contributes to autoantibody formation early in disease. So don't want to spend all my time just on that slide. I'll go to a couple of thumbnail sketch slides of these different projects, and again, try to keep it sort of high level. But we found an interesting variant of this PNP gene. So it's purine nucleoside phosphorylase is the full name, so easier to say PNP. And it's a gene that is actually involved in our cell cycle and nucleic acid recycling. So it's in the salvage pathway, for those of you familiar with the biochemistry pathways, and that's basically a pathway that helps us build nucleic acid building blocks and reuse old nucleic acid building blocks. And so pretty important when our cells go through cell cycle and divide, because we have to duplicate our genome. So there's a common variant allele that's a coding change allele, so it changes the protein structure of PNP. And it's interesting, we found this gene that I've just described impacts type 1 interferon. And it does have an impact on cell cycle. We find that the allele that's linked to lupus is less efficient. And so cell cycle goes forward less well. One of the things we found as we follow that up is that it seems that when cell cycle doesn't work as well, you can leak small bits of nucleic acid into the cytoplasm, or even larger bits into the cytoplasm called micronuclei, where you can see this nucleic acid. We think of it as kind of a dysfunctional genome replication. It looks, we think, like viral particles in some ways to the immune system then, because you shouldn't have all this nucleic acid in the cytoplasm of the cell. And we think it can cause inappropriate activation of antiviral receptors, and that's probably the interferon link. Interestingly, this gene, because it functions in nucleotide synthesis and salvage, it may affect Treg function, because adenosine is a nucleotide, but it's also very important in Treg function. And we're also wondering if the cell cycle issue may interplay with some of our known lupus therapies. As we know, some of our lupus drugs actually block cell cycle and S phase, which is the same phase of cell cycle that this gene variant causes a problem in. So, so that's been a really interesting hit. Like many of them, you get a hit in a genome screen and you don't know what it means, but we've been trying to figure that out, and it's taken us down some interesting roads. With plasmacytodendritic cells, we've actually been interested in these cells, as you could guess, for a while, being sort of interferon-minded lab. As we've mentioned, interferon we think is causal in lupus, and there are a number of unique receptors that can really shut off plasmacytodendritic cells. So these are attractive, I would say, as therapeutic targets, and many people have thought this. There are actually early phase therapeutics directed at the two that are indicated with the arrows to the blue rectangles here, the anti-BDCA2 and anti-ILT7. So there's been some early therapeutic development. I think great minds think alike, and these seem like very logical targets actually for therapy. We've tried to innovate and not just do what everybody else is doing here by trying to think of ways you could target more than one at the same time and develop some synergistic targeting within the cell. In our hands and in our in vitro experiments, we find that ligating one of these suppressive receptors in isolation doesn't shut down the cells that aggressively, and we're wondering if this may result in some partial responses if these were taken forward as therapeutics, that you might need a stronger approach. So we've been working on combinatorial therapies here to see if we could really shut down plasmacytodendritic cells, and that's another thing that's been exciting and interesting, I'll say, in the lab recently, and also interferon related. And then I'd say as the last project I want to highlight, we do have some work on this IRF5 gene, and we've been trying to understand how that impacts autoantibody formation in lupus. I'll say that that's a little bit early on now, but we found the link between the gene variant and the autoantibody, and so now we're trying to understand that. And I just put a kidney biopsy slide here to point out that we definitely see activation of IRF5 in the kidney. We see inflammation, and we think the kidney is one of the organs damaged by autoantibodies in the disease as well. So I think that that's an interesting site to look at. So that's sorry for being a little bit overview on that one, but it's still early days, and we're excited though to think if we could understand how you make autoantibodies. That's definitely understanding a critical, interesting part of lupus that is in some ways fairly specific. As many of you know, the autoantibody can be shared with other diseases, but we still use them diagnostically. So an example would be anti-Rho antibodies are common in lupus, and they're also common in Sjogren's syndrome, but they're pretty uncommon in healthy people. You can find them, but not so commonly. So we think that this is one of these lupus-associated immune system features that would be really interesting to understand better at the molecular level. And then one last thing, we have been working with companies in either the design of clinical trials. We weren't so involved with anifrolumab, but we've helped with some other companies with their anti-interferon drug design efforts. And I think for most of the companies now, it seems like they're buying into this one size does not fit all type of model. I'll admit that that's been a change even within my career, and it's nice to see this shift. And we're hoping in general, some of this work that I've mentioned in brief will help in these efforts. We hope that the genetics and some of these other studies looking at differences between patients may eventually inform how we use drugs, or at least how we do trials, and hopefully get some more robust trial results in lupus. So anyway, thank you for inviting me. It's as I said, I have very warm feelings to the ANRF. You really helped me get started. And I wanted to point out on my last slide, I actually have a junior faculty member in the lab, Teresa Wampler-Muscardin, who I think will be featured in the rheumatoid arthritis series, actually, from ANRF, and she's currently supported. So that's a second generation already of ANRF support. So thank you. I will stop there, and I'll stop my screen share here as well.
Are there any questions?
Okay.
So maybe I have a question then.
Go ahead, Dr. Diamond.
Are there any other questions, Emily? I don't want to hog the.
No, go ahead. No questions yet.
Okay. So, we've all been very absorbed for the past year and a half with COVID. And one of the things that we hear over and over with COVID is that those who have low interferon seem to be at most risk for bad disease. So are there concerns with this therapy with either COVID or any other viral infection? Or what would you be telling patients now about this therapy versus the benefits and the risks?
Yeah, that's a great and very practical question. And I'll say that obviously we don't really know the answer yet. There's some potential risk for the reasons you just mentioned. There were those striking papers in Science last year where they found both knockout type gene variations in the interferon pathway or neutralizing autoantibodies that targeted the interferon cytokine were both associated with severe COVID and deaths from COVID. So that being said, if you look at the trials in anifrolumab, it seemed like very particular viral infections were greater risk. There was some herpes virus for example, and this seems to be the case with a lot of our biologics, which is not well understood. I know many of you know this, is that we roll out a biologic that blocks a major immune system pathway, and we end up getting some very particular infections, but not all infections are necessarily increased in risk. So whether COVID actually would be increased in risk with anifrolumab, I think is a question, but it's a fair question. again, one I don't know if we'll know until the human experiment would play out. So there's some risk there. Obviously, vaccination is really important, and that will help your defenses for sure. Sorry for getting on the soapbox there for a minute, but it's super important.
You are
no matter
so
whether
right.
whether you take anifrolumab or don't or whatever, everybody should do it. And that could help you. And a lot of our other therapies too, it's just tricky. So I don't know if there will be, I think the verdict is still out whether anifrolumab would be particularly more risky. Luckily, our big registry studies have supported that a lot of the other lupus therapies don't seem to pose a lot of additional risk within COVID. And so I think, but I agree with Betty that, this is hitting a pathway that's shown to be important in our defenses. So I think, hopefully the pandemic won't be with us forever and this won't be so much of an issue, but I think for right now, it's a question that's really important and we don't know the answer yet.
So thank you. This brings us to our next speaker. And our next speaker is Jason Knight, who received his MD and PhD from the University of Michigan, where he remained for clinical training and is now an associate professor. He was a fellow of the ANRF from 2016 to 2018. A major interest of his is vascular disease in lupus, and he has become very interested in vascular disease in COVID and in the mechanisms of vascular disease in both lupus and COVID, differences and similarities. So it's terrific to have another a graduate who's very involved in very topical work. So, Jason?
Thank you, Dr. Diamond, for that nice introduction. Let me share my screen. This looks like a talk. Yes, okay. And thank you also, Dr. Diamond, for giving me feedback on words I had written on that very topic just a few hours ago this morning. So you are doing a lot of things all the time. Yeah, truly been doing a lot in the COVID space, but today I'm going to talk a little more about my main passion in the area I hope to keep working for the next 20 and 30 years. COVID, I hope we can eventually retire that work. And so my favorite thing to study is this lupus-associated condition called antiphospholipid syndrome. Sometimes we diagnose it in its own right and we call it primary APS, but it commonly complicates lupus. I think it's really relevant to lupus, living with lupus. There have been a few attempts at international criteria. There's a third one underway now that I think is going to be a little better. Maybe this will be out next year. We should look for certain lab tests. These have clunky names that are difficult to teach to our trainees, but we're kind of stuck with that. But geez, the sad thing is the clinical criteria. So, to make this diagnosis, we really require something bad to happen to you. We may know the antibodies are in your blood, but we typically don't start treating preemptively until there's a blood clot or one of these really devastating pregnancy losses. And as I mentioned, you're going to find at least the antibodies in, maybe a third of patients with lupus. And if these antibodies are in the blood, we know it predicts a more severe lupus disease course. So I think really relevant to lupus as well as people with primary APS. So how do we get here? Well, you can make a quick argument that United States of America would not exist without lupus and APS. And so to do that, you need to think about line of succession of the British monarchy. From Hamilton the musical, we're all familiar with George III, who did not get along well with the colonists, leading to them declaring their independence. And you can contrast him with Anne Stewart, who by all accounts was a great unifier of people, brought England and Scotland together for the first time under her reign. But why this transition from House of Stewart to House of Hanover? Well, the problem, or a big part of the problem, was that Anne's health was not at all good. Historical record seems quite clear that she had lupus. But then there's also these features that look sneakily like what we see in our APS patients, where seizures are very common, strokes are certainly common. She ultimately died of a stroke. But if that doesn't convince you, then you look at her pregnancy history, where 17 times she tried to make an heir to the throne, and all ended in stillbirth or death in infancy from complications of prematurity. So she did not have a good body for bringing an heir into the world. And so if not for Queen Anne's lupus, you might wonder if I'd be giving this talk from Ann Arbor, United Kingdom today. So what is a phospholipid? This clunky word that I have to write a lot of times in a grant. So it's 1 part water soluble, 1 part fatty. And it kind of looks like this. And it turns out that these phospholipids are really good for building membranes. You can make this membrane around the cell, keeps the inside parts in and the outside parts out. And we all know about antibodies. Antibodies, do good things like neutralize infections, but in the case of APS, we get antibodies, either to the phospholipid layer directly or to proteins that are closely interacting with it. And so this damages cells, activates cells, and causes problems. So I decided to just highlight a couple of the stories that we're presenting at the ACR meeting this year. So this is all kind of newer stuff that's not published out there anywhere. And so this first story is by AJ Tambrali. He's a good candidate to maybe be a future ANRF scholar. I also have one ANRF scholar that I'm mentoring in my lab. So it's just great to see how the support is for young investigators. But so AJ has been studying neutrophils. This is one of the cells we like to study in the lab. This is a type of white blood cell. It's really abundant. I mean, we all say that our stuff is understudied, so I will say the same. I think it's still relatively understudied compared to other types of cells in lupus and APS. One of the things neutrophils can do is release these structures called neutrophil extracellular traps or nets. And so this was first described in 2004, although presumably neutrophils have been doing this for a lot longer than 2004. And so these are spider webs that get shot out into the world, get shot out of the neutrophil and create a web much bigger than the original neutrophil. And these webs have antimicrobial proteins in them, so proteins that can kill bacteria and maybe viruses and maybe funguses. Very interestingly is that the web is built of DNA. So that DNA had been super compacted in the nucleus, but it gets uncompacted and shot out into the world. And that's the main backbone of the webs. And then there's other proteins like histones that are also common in nucleus. So nets do a lot of good things, presumably. They help us stave off infections, but with their power comes responsibility. And it turns out that it was not long in this field before it was realized if you release a net into the bloodstream, it's a very good way to trigger a blood clot. And so we know APS has a lot of blood clotting, and that's one of the reasons we started to look at this. And so this story, just in one more minute, is based on some work we did a couple years ago where we looked at gene expression of APS neutrophils and how that compared with healthy neutrophils. What made these neutrophils program differently? And one of the kind of key pathways, so here the supercomputer has looked at all the different genes and has helped us understand what themes exist there. And one of the themes was for metabolism. And if you look at the higher level words, the wonkier words, it turns out there's a lot of pathways that involves the utilization of glucose. And so we thought it might be interesting to see how neutrophils use this sugar glucose. One of the ways that cells use glucose is through this process called glycolysis that you surely learned about in high school biology. This is a very fundamental way that a cell can turn a sugar into energy. And so if, there's certainly a spread, there's variability, each APS patient is different from the next, but there's, on balance, it looks like these APS neutrophils are programmed to be very hungry for that glucose to really power through it. And we even have some preliminary data in an animal model of APS where if we transfer in a glycolysis inhibitor, we can really effectively shut down blood clotting. So APS mice get very big blood clots and we're able to push those down to control levels with an inhibitor. And then the other project, and this one will be a little faster, this I'm very excited about. We just submitted a grant yesterday on this topic, so it has been on my mind. But the idea is that APS patients can have this skin change, commonly have it, a third to a half of the time, a skin change called livedo reticularis or livedo racemosa. And this is a sign that the blood flow in the skin is having trouble, that it's gotten too sluggish. And the interesting thing is if you have this on your skin, it predicts there's things going on inside of you, that either the brain or the kidneys or the heart, that there can be issues with these other organs. And so, the idea is, can we, as we learned in also in high school biology, skin is the largest organ in the body. So, it has stuff like blood vessels and nerves in it. And so the question is, can we profile the blood vessels in the skin to give us a window inside APS where we don't have such easy access? And we can use this fancy and expensive technique called single cell RNA sequencing to biopsy the skin, make it a single cell suspension, and then we can like barcode and study gene expression in each of these individual cells. And so, here we can see endothelial cells, which are really important blood vessel cells. They line blood vessels and interact with the blood. The blood is always trying to clot, and these endothelial cells are always trying to stave it off. So if they get regulated poorly, things go bad. I guess the hypothesis or the idea we're following is that endothelial cells are kind of the starting lesion. They are the ones interacting with the blood where the anti-phospholipid antibodies are living. But then there's lots of interesting potential for them to communicate with other cell types, both in the blood vessel wall, but also in the surrounding tissues. And we think this can potentially lead to some remodeling over time. If you see errors here, do not tell me, because I put this in Grant yesterday, but this is just to give you an idea. We think factors coming out of these microvascular endothelial cells can then be feeding back onto the cells themselves, but also into other cells and causing problems both locally and maybe even systemically. So that's just a couple of highlights of what you'll see at ACR. Again, I just want to say so much thanks to the ANRF for supporting me at a very important time in my career, and thanks for the invitation today.
Thank you. Are there questions for Jason?
Nothing that's coming so far, but we'll keep an eye out.
Okay, I do have one, but maybe I'll keep it because we're getting a little behind schedule, I think. So the next speaker is Jolene Cerman, who received her Ph.D. from the University of Leiden and performed postdoctoral research in my laboratory and was an ANRF fellow from 2019 to just 2021, this year. And she's been studying ways to stratify patients with lupus. Tim mentioned that we don't think that interferon blockade or any other therapy we currently have for lupus will work in all lupus patients. And as she'll tell you, one of the reasons for this is we think there's heterogeneity of disease. So that's what she's exploring. So Jolene.
Thank you, Betty, for that kind introduction. So I think you can all see my screen. Go to the first slide. Okay. So yes, as Betty mentioned, today I would like to talk about the way that we can group lupus patients based on the B cells that are present in these patients. And so this is work that I started actually when I was in Betty's lab. So lupus, as probably most of you know, is characterized by autoantibodies. So these antibodies target our own tissues, and most common are so-called ANA autoantibodies, and these bind to nuclear structures in the cell. And these antibodies can then bind and cause inflammation in various tissues. And in the end, this can lead to organ damage. So antibodies are produced by plasma cells. So the plasma cells are kind of the factories of antibodies. And these plasma cells arise when there are precursors to B cells are activated. And so it's kind of this sequence of events that leads to the production of antibodies. So what my work previously has shown that there are actually different types of B cells and plasma cells in lupus patients. And this is in particular true when we look at these ANA positive cells, so the cells that actually make these antibodies. And so what we observed was that there were two groups of patients that, just for simplicity, I will call it type 1 and type 2. And so these patients present with different types of B cells. And so our idea is that we can use this to actually group patients and see if we can use specific therapies in these patient groups to target just one of these activation pathways. Okay, so this is also called precision medicine. So I think Tim Niewold also mentioned this earlier, that we want to develop ways that we can take apart the heterogeneity of lupus. So here you can see a whole group of lupus patients, but these patients, we think that they are actually quite different. And so I focus here on these two types that we identified, but there are probably more different types. And what we want to do is that we want to take apart these patients and then see if we can use specific treatments for each patient groups. So in order to develop this, what we need to do is we need to know what specific treatments can be used in these patient groups. And we also need to be able to accurately take apart these patients and divide them into the groups. And so that is what I've been working on in the last few years. And so I will first focus on the specific therapeutic targets that we have been looking at. So what we have been doing is we have been using a technique that Jason Knight, the previous speaker, also talked about, the single cell RNA sequencing. So this is a technique that you can look at all the molecules that are present in a cell. So in this plot, every dot is a single cell. And we looked specifically at plasma cells. So these are the cells that make these autoantibodies. And what you can see is that the cells and how they look are different between the two patient groups. You can see, for example, in type 1, there are many cells of the purple and the green phenotype, whereas in type 2 patients, we see actually more of these bluish cells. And so that means that there are actually different types of plasma cells that we think that we could maybe target therapeutically. Then next, we also looked at these cells and to see if we could actually identify different molecules that could be therapeutically targeted. And so we identified several molecules. I just show a few examples here. You can forget about their names, but I just want to show that we can actually see molecules that are present very frequently on cells of a particular type. And so we think that we can now actually look at these molecules and see if there are, for example, existing therapies that can already target these molecules, or that we need to develop new medications. So the second thing that I mentioned is that we also need to be able to accurately identify in which group the patient falls. And so far, we have been looking at these B cells, and this is not a very simple, accessible way to separate the patients. And so to apply this in larger clinical trials, as well as in clinical practice, such as hospital or outpatient clinics, we need, or we would prefer to have a simpler assay. And so we have been looking at the ANA autoantibodies that are present in these patients. And so we have previously shown that patients with type 1 have actually more different antigen specificity, so more different ANA antibodies, whereas the patients with type 2 generally have higher affinity antibodies. So these are antibodies that can bind stronger. And so we now actually wanted to study this in larger patient groups. And this is what we have done here. And actually our main finding was that the autoantibodies are also different in the type of autoantibodies that are present. So quite commonly in lupus, there are RNA binding proteins that are recognized by these antibodies. And so there are the four main ones here. And what you can see that patients with type 1 very often, so this is about 3/4 of the patient, have antibodies that recognize one or more of these RBPs. And in particular, the difference is large for patients that recognize 2 or more of these proteins. We are, of course, further developing this because it's not a black and white difference. So we are hoping to find more specific correlates of the B cell phenotype. But I hope that I convinced you that it is important to take apart the heterogeneity in lupus and that we can actually identify which of the B cells or plasma cells are actually causing the damage. And then we want to use that to prevent and block these specific cells to have more effective treatment and less side effects. So of course, this work could not have been done without many other researchers and organizations. So as I mentioned, this work started when I was in Betty's lab, and many people of the Feinstein Institute were also involved. And some of my more recent work has been done by a PhD student in our group, Hugo van Doren. And of course, I'm very grateful to ANRF for funding me in this early stage of my career. I would be happy to take any questions.
So are there questions for Jolene?
There is a question, Dr. Diamond. The question is, can someone have lupus and rheumatoid arthritis at the same time? And do the above conditions have similar symptoms?
So I have to admit, I'm not a medical doctor, but so probably Betty can answer this better, but I do think you can have both at the same time. And some of the symptoms can be in common. So in lupus, you can have arthritis, right? But I do think there are, in terms of, for example, the antibodies that I talked about, there are actually quite specific differences, but there are, I think, overlaps when both can be present at the same time. But Betty, please correct me.
No, what you said is absolutely correct. In the antique days when I was in medical school, the overlap was called rupus. There are different antibodies. There are some different susceptibility genes. On the other hand, there are certain commonalities. And you know, I think as we learn to understand some of the commonalities, we'll learn more about what therapies used in one can translate to a particular subset of patients with the other disease, and vice versa. So it's an important question. So maybe, are there more questions, Emily?
No, that's it.
Okay, so I'm going to go on then, because we are running out of our time here. And our last speaker will be Sarah Baxter, who received her MD and PhD from the University of Washington in Seattle and stayed in Seattle for clinical training, and is a current ANRF fellow. And we're circling back to an interest in DNA, and what you heard at the very beginning in the first talk was how the RNA made from DNA can be made into different transcripts that make different proteins. And Sarah is interested in enzymes that actually can cleave DNA. You actually heard from Jason's talk that sometimes you find that outside of cells. So Sarah.
I think you're muted, Sarah.
Thank you so much. I was just saying, it's such a joy to hear all of the successful scholars that have previously come before me, and I'm so grateful for the ANRF support. So I'm a pediatric rheumatologist, and I'm also going to be talking about the genetic diagnoses of pediatric lupus, actually. So for many kids, rheumatologic diagnoses have genetic origins, and I'm so grateful that some of the earlier speakers spoke about this a little bit. To put a finer point on what I'm looking at is there are common mutations that you find by looking at whole populations of children or adults with lupus that have some effect on your risk of lupus. And then there are causal mutations. So people who you can say have a defect in a gene that caused their disease directly, didn't just slightly contribute to the risk. And so those are sort of common variants versus rare, severe variants. Both are important, both absolutely contribute to lupus. And so I'm studying the latter. So oftentimes we found that establishing the diagnosis can improve care. And I just wanted to actually start because as a practicing pediatric rheumatologist, I see patients, and that is what drives me. So this is one patient, little 3-year-old girl. She presented, she had swollen lymph nodes, she had swelling around her eyes, and her labs essentially showed lupus. But because she was 3, which is very young for lupus, we were able to get clinical genetic sequencing. We were able to diagnose her with a genetic disease, with a mutation in the gene NRAS. And because of that, we were able to direct her care and get her not only appropriate medical care for her lupus, but we're also screening her for cancer, for which she's at much higher risk. A sadder opposite story is an 11-year-old that we treated at our hospital who had autoimmune bowel disease, autoimmune cytopenias, very much like our lupus patients, but because of her age, because of her presentation, because of the time that she presented, only underwent single gene testing, and that didn't show anything. So she had the standard care, which was just immune modulating therapy. And unfortunately, she later passed away of infection. And after passing away, broad sequencing showed that she also had a genetic disease, a mutation that was so severe that it drove her presentation, and it actually would have changed her care. This is a child who would have gone to transplant. So in my prior work, before coming to the ANRF in my lupus work, I looked at 123 very young children who had multi-system autoimmunity affecting lots of their bodies. Sounds a lot like lupus, of course, which is my first love. It's called IPEX-like syndrome, and there had been no mutation found in one gene. But of course, as opposed to 20 years ago, for any given patient, we can look at 20,000 genes, not just one. So within these 123 small children who had autoimmunity affecting their whole body, we found that nearly 40% had a genetic diagnosis, a single gene that had such a severe mutation that it was driving their disease and actually would have changed their care. So 42 of those 48, you can see this larger dark blue wedge of these patients would have had a change in their treatment. So for this IPEX-like syndrome, this multi-system autoimmunity, we've really found out it's important to sequence early, sequence often, and sequence broadly. And I want to get there for lupus, and that is my goal. So current treatment for lupus, as you've heard, is really trial and error with medications that we know well until something works. And you heard our last speaker talk about precision medicine, and there are lots of different ways about it. And I think that there will be many different ways to get towards precision medicine. I'm interested in genetic diagnosis, which allows us to look at the basic biology that is driving a patient's disease and then match that treatment to that biology. The way that I just think about lupus is a snowballing reaction. So there is at some point a break in tolerance. Our bodies get confused and start reacting and hurting ourselves. And then that inflammation, that damage then compounds and snowballs downhill, and you end up with full body lupus, autoimmunity, multi-system. And I'm interested in that initial trigger that starts that snowballing reaction. But to find those triggers, to make these genetic diagnoses, we have to start somewhere. And where we need to start is we need to know what mutant genes and pathways cause lupus, how those genes and pathways differ clinically from each other, and how to treat those different genetic diseases. So what we do in my lab is we go family by family. And so this is one such family that I'm studying with the ANRF. And the circles are girls and the squares are males, and horizontal lines are marriages and vertical lines are progeny. So the circle at the very bottom is my patient, a young girl who has childhood onset lupus. And you can see her mother also had childhood onset lupus, and her mother had childhood onset lupus. So 3 generations of childhood onset lupus, which we think is probably a stronger genetic effect as opposed to people who develop lupus more because of a microbiome or environmental or polygenic source. So what we did with this family is we sequenced them and we said, what is different between the patient and the family members who do not have lupus, and what is the same with those who do have lupus? And using computational analysis, we were able to find a mutation in AIM2, which looked damaging if you look at the in silico predictions. If you look at what the computer says should be conserved and probably hurts the protein. But finding this and thinking it looks like a good target isn't enough. So what we did is we looked around and we found, the first thing is to try to find other patients with lupus with the same mutation, or at least mutations in the same gene. And we did find 2 other individuals in our cohort of over 300 patients with lupus who had mutations in the gene AIM2. Different mutations, but 3 different individuals, all with lupus, all with mutations in the same gene. But again, that's not the whole story. We need to show that these mutations actually change something. So right now what we're doing is we're characterizing and validating AIM2. So what AIM2 does is it recognizes free double-stranded DNA, which lupus patients will probably recognize as that anti-DNA DNA that your doctors always talk about in your appointments. So AIM2 actually recognizes that, and when it sees that, it induces an inflammatory response. So what we're doing is we're saying, do these mutations change the function of AIM2? Do they change the function of its ability to recognize double-stranded DNA? And do they change its ability to cause a downstream inflammatory response? So in order to do that, we're looking at the RNA, and we've shown that all variants are expressed. We're looking at the protein stability, and we've seen that one of these proteins actually is not stable. The other 2 are. And now we're looking at the binding. So can AIM2 bind with its partners, and can it actually assemble along with its partners this inflammasome, which is an inflammation-making machine. And we can make that inflammation-making machine, that inflammasome, in a cell culture in a dish, and then we can go back to the patient's original cells, stimulate their AIM2, and look at how their original cells work as well. So really taking the gene all the way back and saying, how is the function altered and how does that affect inflammation? This is not the only patient. And so, my goal, I have over 300 pediatric patients with lupus, and over 60 of those we have trios on. And these are just a few of the families, and some families have a strong family history and other families don't, but every family really has a story to tell. And by doing deep sequencing, looking at that family level, we're interested in those severe mutations. And then finding those severe mutations can point us towards genes that will be more important in GWAS studies and looking at polygenic risk scores, or in looking at treatment options. So my eventual goal is really that every child with lupus receives a genetic diagnosis so that they can benefit from treatment, precision treatment. And thank you to everyone. I work in the lab of Mary Claire King, who revolutionized breast cancer research about 30 years ago, and is excited to turn her attention to lupus and help us bring genetics to children. Thank you to all our patients and families, and thank you to the ANRF.
So I would like to thank these speakers and audience. I think that what you see is the talent that the ANRF has identified, and that lupus research has a bright future, that there are many avenues to explore, that our graduates have already brought some clinical research into clinical practice, and we're hoping to see that from the more recent and current fellows of the ANRF. This has been a terrific session, so thank you all.
Thank you, Dr. Diamond. Thank you all to our speakers today. As Dr. Diamond said, I think we can all learn from this incredible research and be so proud of all of the work that our scholars and alumni are doing. We hope you can join us for our next Spotlight series in 2 weeks on rheumatoid arthritis. Look for that link on our website. Thank you again, and have a wonderful day.